Welcome to this explainer. Today, we're diving into what is honestly a fascinating medical detective story. For the longest time, the exact mechanics of cancer were just totally shrouded in mystery. But today, we're going to decode the genetics of cancer. We're going to explore how tiny, microscopic errors in our DNA can cascade into a major disease, and, more importantly, how scientists are now using that exact same genetic code to actually fight back. So, let's jump right in. Here's our roadmap for the explainer. First, we'll cover cells, division, and mutations. Second, cancer as a genetic disease. Third, proto-oncogenes and tumor suppressors. Fourth, driver versus passenger mutations. And finally, number 5, the promise of precision medicine.
Okay, Section 1, Cells, Division, and Mutations. Let's start with the basics. To really understand what goes wrong in cancer, we first need to establish what normal looks like, our status quo. Our bodies are made up of trillions of cells, and they're constantly dividing for growth, for repair, for general tissue maintenance. I mean, think about this. Right now, literally millions of your cells are dividing every single second. It's an absolutely awe-inspiring scale of constant biological machinery. But when cells divide, typos can happen in their DNA. These are mutations, and it's crucial to understand the difference between two main types of these typos, germline and somatic. Germline mutations happen in reproductive cells, so the egg and the sperm. If a mutation happens there, it gets passed on, meaning it'll affect every single cell in the next generation. Somatic mutations, on the other hand, occur in your non-reproductive body cells, the soma. These aren't passed on to your kids. Instead, they only affect the descendant cells derived from that specific mutated cell inside your own body. Because of the sheer volume of cell division happening inside you every day, your body is naturally going to acquire these somatic mutations as you go through life.
Moving on to Section 2, Cancer as a genetic disease. Let's look at what happens when those mutations build up. What you're seeing here is a very clear shift from order to complete chaos. Take a look at the normal cell. Notice how it goes through a highly controlled cycle of division? It's tightly governed by regulatory signals. Basically, it stops dividing when it's told to stop. But then, look at the cancer cell. Those regulatory signals? Completely ignored. The cell cycle just loops wildly out of control, multiplying into a chaotic mass of abnormal cells. Cancer really isn't just one single disease. It's a whole collection of related conditions that are all driven by this exact type of uncontrolled growth. When that genetic engine breaks down, we see a few core characteristics emerge. First, as we just saw, is unregulated rapid cell division. Second, these cancer cells gain the terrifying ability to evade normal programmed cell death. They basically dodge the natural mechanism that's supposed to destroy damaged cells, kind of making themselves immortal. Third is metastasis, which is where these cancerous cells break away from their primary site and spread to totally different tissues and organs. And finally, the root cause of all this chaos. It's driven by the steady accumulation of those acquired somatic mutations over time. This whole devastating combination of rapid growth, immortality, and spreading is exactly why cancer is so incredibly difficult to treat and why early screening is absolutely vital.
Section 3. Proto-oncogenes and tumor suppressors. Think of these as the accelerators and brakes of the cell.
Let's start with proto-oncogenes. In a normal, healthy cell, these genes encode growth-promoting factors. They basically stimulate cell growth and division. A great way to think about them is like the gas pedal in a car. You need the gas pedal to move forward, to grow and repair tissues. But you only want to press it when it's actually necessary. On the flip side, we have tumor suppressor genes. These genes produce growth-inhibiting factors. They hit pause on the cell cycle to repair damaged DNA, or they even induce programmed self-destruction if the cell is just beyond repair. If proto-oncogenes are the gas pedal, tumor suppressor genes are your crucial brakes. You absolutely need them to stop the cell from dividing if something starts going wrong. So if we trace the path from a normal cell to a cancer cell, you can see how mutations act as the spark. A mutation to the proto-oncogenes causes a gain of function. That's effectively like jamming a brick onto the gas pedal. It gets stuck pushed all the way to the floor. Conversely, if a mutation hits the tumor suppressor genes, it causes a loss of function. It essentially cuts the brakes. The really important part here is how these mutations actually take effect. Gain-of-function mutations in those gas pedal proto-oncogenes are dominant. That means it literally only takes one mutation, just one hit to one allele, to jam the gas pedal and drive uncontrolled division. But loss-of-function mutations in the tumor suppressor genes are recessive, and this brings us to what's known as the two-hit hypothesis. Because you inherit 2 copies of every gene, both copies, or alleles, must be mutated and inactivated to completely cut the brakes. The cell needs 2 hits to fully lose its ability to stop growing.
That brings us to Section 4, driver versus passenger mutations. Let's talk about tracking that progression.
So you might be wondering, do all of the mutations that a cell acquires actually cause the cancer to grow? The short answer is no, definitely not. As our cells divide over our lifetimes, they pick up a ton of genetic typos. But we categorize these into 2 distinct groups: driver mutations and passenger mutations. Driver mutations are the active threats. They're the key genetic alterations that directly contribute to cancer development by giving the cell a specific growth advantage. Passenger mutations, on the other hand, well, they're just along for the ride. They are completely neutral genetic changes that just happen to accumulate right alongside the driver mutations, but they aren't actively promoting the malignant growth.
Let's trace how this looks on a timeline. Those gray asterisks you see represent passenger mutations, just accumulating harmlessly over the years. But the red asterisks? Those are your driver mutations. Notice how cancer arises step by step as those specific driver mutations stack up, granting the cell new, dangerous abilities, like a growth advantage or the ability to metastasize. You'll also notice that both our inherited background, things like hereditary cancer syndromes, and environmental exposures like carcinogens, act as modifying factors. They can accelerate this entire timeline, speeding up how fast you accumulate those critical driver mutations.
Finally, Section 5, the promise of precision medicine. This is the future of treatment.
And now we arrive at the most exciting part of this whole explainer. We understand the problem, so let's look at the solution. How do we actually stop this broken genetic engine? I mean, if cancer is driven by specific genetic mutations jamming the gas pedal and cutting the brakes, how do we go in and fix it? In traditional treatments, like standard chemotherapy, the goal is often just to broadly kill rapidly dividing cells, which, as you probably know, can cause significant damage to normal healthy tissues too. But what if we could use the cancer's own unique genetic code against it?
That exact question is the foundation of precision medicine, and it follows a highly logical process. Step 1, doctors sequence the DNA of the individual's specific tumor. Step 2, they analyze that unique genetic profile to identify the specific driver mutations. They find exactly which genes are jammed or broken. And Step 3, rather than just relying on broad treatments, they tailor very specific therapies to directly disrupt those mutated molecular pathways, which minimizes the side effects on normal tissues.
Seeing this workflow in action is pretty amazing. The first step involves comparing the DNA sequence from normal cells with the tumor cells. We filter out all the noise, all those passenger mutations, so we can isolate the key driver mutations. And once isolated, the treatment is designed literally like a key for a specific lock. It targets only that driver mutation with a specific drug to halt the uncontrolled growth. It is incredibly elegant science.
Now, of course, we have to be realistic. The clinical outlook isn't without its hurdles. Tumors are incredibly complex. We face challenges like tumor heterogeneity, which means that not all the cells in a single tumor even have the exact same mutations. And cancer can unfortunately develop resistance to these targeted drugs over time. But even with those hurdles, precision oncology represents a highly promising approach. It completely shifts the paradigm from treating the organ where the cancer started to treating the very specific genetic errors that are actually driving it. So I'll leave you with this final thought. We've seen how cancer essentially exploits the very code of life to multiply and spread. But by decoding that unique genetic blueprint, we're finally learning how to turn the tables. If we can do this, how close are we to a world where cancer isn't an automatic terminal diagnosis, but instead a managed, chronically targeted condition? It is a deeply hopeful future, and it's all built on understanding the profound machinery inside our own cells. Thanks for joining me for this explainer, and keep learning.